welcome back, ladies and gentlemen. So now, are you ready for the first session? Are you ready? Yeah, okay. So I've told you that according to the genetic variations, you know that's a particular drug that might be really effective for most people, but in some others they might not be effective at all, right? So we will see if we use the genomics and personalized medicines, how would it be? With this topic of how genomics and personalized medicines can transform healthcare by Professor Rory, Medical Director, Healthcare UK. He is also the Professor of Respiratory Medicine and Honorary Consultants in Respiratory and General Medicine, Imperial College Schools of Medicine's Hammersmith Hospital in London. So ladies and gentlemen, please join me to welcome Professor Rory Cho.
Thank you very much. It's a great honour and pleasure to be with you this morning. I'm here representing Healthcare UK, which is an organisation created by the UK government and funded by UK Trade and Industry and Investment, the NHS England and the Department of Health. I am a respiratory doctor. I've spent much of my life seeing patients in the clinic, using a stethoscope, using a chest x-ray and prescribing medication. But I, like everyone in this room, are now aware of two new technologies which are coming into medicine, which are very different and will change the way we all work. Genomics, the knowledge that we will gain from the understanding of the human genome and the mutations in the human genome, and informatics, the ability to handle very large amounts of data and get answers from huge amounts of information that are not, as individuals, we cannot possibly analyze. I'm going to suggest that these are going to revolutionize our understanding of disease, the process of drug discovery, the diagnostics we use, and our treatment of patients. What I think is going to happen is that we're going to move from a position where we doctors rescue people with serious illness to a position where we can anticipate illness, identify risk, and help people prevent illness. The challenge we have is how we take all this knowledge and new technology and provide benefits to all our patients. So we in the UK have been at the forefront of this area of science for well over 50 years. The structure of DNA was identified in Cambridge, in England. Many of the basic technologies were invented in our country. Professor Southern invented the Southern blot for separating DNA. The basic technology used by Illumina in whole genome sequencing was invented in Cambridge. We have played a key role in the initial decoding of the human genome, and we are currently embarked in one of the largest translational medicine programs in the world, doing whole genome sequencing on 100,000 samples from 70,000 patients. And I'll say a few words about this last project in a moment. We also have a tradition of undertaking large translational medicine programs. This particular program, the Biobank program, started about 5 years ago. We have recruited over 500,000 people, collected complete information on their medical history, a broad range of biological samples. All patients are having genome-wide associated analysis, and we're following them up for over 10 years in an attempt to predict if there are any mutations, identify any mutations which predict what happens to them over that 10-year period. So we have a track record of doing very big studies. We have now started what is called the 100,000 Genome Study. This study is specifically focused on people with medical problems that exist today, on people with rare diseases, with presumed monogenic diseases,
and cancer. The logistics of this study have been very challenging. To do whole genome sequencing on 100,000 samples generates a huge amount of data, a huge number of samples, and it's a very complex process. It has required substantial development in infrastructure. So, for example, we have built a biosample center which is run on a robotic basis and can handle 18 million samples. We have built a sequencing center which is able to do many hundreds and thousands and up to several thousand samples a day. And importantly, we have a data center to handle the data. Each whole genome sequence produces a terabyte, at least a terabyte of data that has to be handled, compared with the clinical features, and compared with the genome sequence of many other people who do or do not have the condition that is being investigated. Structurally, we have created an organization called Genomics England. This is a company owned by the government. It organizes the process, manages a broad range of subcontractor companies in order to deliver the project. It has a clear objective to generate the answers to the questions, which mutations contribute to which diseases, but also to lead into the use of this technology in future in clinical practice. It also has an agenda to stimulate industry and commercial organizations can form partnerships with Genomics England in order to develop new diagnostics, new technologies, and new pharmaceutical molecules. The question is, how do you take all this and actually help patients in the clinic across a country of over 60 million people? Well, we have an advantage in the UK because we have the National Health Service, or NHS. And the NHS is the system where all patients get their care in the United Kingdom. It has been compared by an organization in New York, nothing to do with the UK, called the Commonwealth Foundation, and they looked at our health system and many other systems in developed countries, and they gave us a very high score because for quality, for access, and in particular for cost effectiveness. So we seem to have a good universal system. How do we help that deliver benefits for patients using genomics? Well, what the UK has done is created a structure with genomic medicine centres in each of the different parts of the country, serving as a focus for research, for clinical service, and for training. And each of these genomic medicine centres serves a population of around 3 to 5 million people. Each centre brings together the universities in that area and the big hospitals and creates a central arrangement whereby all the clinical and academic people can work together and serve their local community, but where these centres can work in a network and share resource across the country. So, for example, this is a sign that is up in the clinic in here. And this is a lady who goes to the clinic with her little child, has a test, and goes home. And a few days later, she gets a telephone call from the specialist in this clinic here saying, hello, they have a conversation and tells the lady that the result is that her child has phenylketonuria, but the country's experts here will be able to coordinate the care through her local centre up here. So in this way, she's getting local care, but it's coordinated by the national experts. Similarly, where we have a very rare disease like retinoblastoma, we have 2 national centres, one in Birmingham, one in London, but they have connections through the regional centres so that all the patients in the country where there is a retinoblastoma can have their care supervised by 1 or 2 national experts. And we are now evolving that to take advantage of the new knowledge. So, for example, when I was a doctor seeing children many, many years ago, if a small child presented with diabetes, we assumed that insulin was going to be the correct treatment. We now know that actually neonatal diabetes has 5 subtypes based on the genetics of the child, and that only one of those 5 subtypes requires treatment with insulin. This is very rare, but the specialist centre for this is in the southwest of England, and wherever children across the country are identified, through working together across the network, the expertise from the group in Exeter can be delivered to the patients and their families. The whole genome sequencing project is also delivering benefits to patients. Here is an interesting example. We always think about monogenic disease as a problem that's identified early in life. But here we have 2 patients who are actually in their 70s. They presented in their 50s with a neuropathy, with neurological problems in their legs. They're brothers. There's a history of similar illness in the family. They'd had a range of tests which had not been able to identify what could be the cause of this. And eventually, in their 70s, their samples were analyzed by whole genome sequencing. And a mutation was found in a gene associated with the transport of the amino acid serine. This was found in both brothers. And it's quite interesting because now that opens up a possibility that maybe if we can give these people high doses of serine in their food, maybe that can help with the difficulties of local transport within the cells. So it opens up not merely a diagnosis, but also the opportunity of trying out approaches to treatment. We're also seeing the development now of specialist clinics for inherited disease. So, for example, at the Brompton Hospital in London, they have a clinical service specifically targeted at inherited cardiac disease. You will know that particularly hypertrophic occlusive cardiomyopathy is an inherited cardiac disease associated with sudden death, particularly in athletes in their 20s and 30s. This is a genetic disease. We know what the gene is, and if it's identified, preventative therapies can be instituted, and this sudden death in these young people can be prevented. This disease is an interesting example of how we can catch the public's interest in this area. Unfortunately, quite a famous man in the United Kingdom died, and he had at the time of his death, the autopsy identified that he had this condition, but he'd never been an athlete and it hadn't caused him any problems. Nobody noticed it, but a few years later, his son, who was an athlete, dropped dead at the age of 31. And that shocked people, and so a large number of people have raised money, and here a group of people cycling on a long-distance cycling trip to raise money through charity to help the research into this particular condition and to make people aware of the risk of having this genetic abnormality so that families and individuals can be tested. This is in fact the panel that is used at Oxford in England to test for inherited cardiac conditions. This is the current panel. And it looks for mutations associated with dilated cardiomyopathy, hypertrophic cardiomyopathy, left ventricular non-compaction, restrictive cardiomyopathy, and Wolff-Parkinson-White syndrome. And so where families have a history suggesting any of these conditions, they can be tested, and their children can be tested, and preventative measures can be put in place. I was at a meeting a few weeks ago with a cardiologist from China who was saying that actually in China, they probably have at least 1 million people who have the mutation for hypertrophic cardiomyopathy. So this is potentially quite a big problem for people, for countries to think how to deal with. So I've talked a little bit about how already we're starting to make an impact on early identification of monogenic inherited diseases, and to start to think about strategies to identify people early and to get them into clinical pathways to prevent complications. And before finishing that, I want just to emphasize that last part of the sentence. It's quite easy to have a test. The test is really only a start. What's important is what happens over the rest of that person's life and how we look after them. We're also looking at cancer. And what we and everybody is finding is if we take samples from cancer and do the genomic analyses, we find a large number of mutations. And we're able then to look at the classification of the cancer using these mutations and subdivide cancer into subfamilies. And that's really important because traditionally we've given all patients with a particular type of cancer the same treatment. But it may well be now that each subfamily has to have specific precision treatment that actually is targeted exactly at those patients with those specific mutations. And I'm just going to show you a couple of examples. So pancreatic cancer, as many of you will know, is a very, very serious disease with a very short life expectancy. Recently, we've had a breakthrough in that we've been able to identify, using a genetic analysis, that that there are really 4 types of pancreatic cancer and that they are distinct. For example, the squamous type has a life expectancy much shorter than the others. So now this opens the question. Previous treatments of pancreatic cancer which have not been successful may well have not been successful because most of the patients in the study group had the wrong subtype. So we now have to go back and look at pancreatic and indeed other cancers and think about medication which is specific for each subtype and have a review of the clinical studies using that idea. I just wanted to show you this recent study because I think although it is just one study, I think it highlights this whole issue very clearly. There will no doubt be more studies on this issue because I think it does open a whole new door to the way we're going to look at cancers. So this group looked at patients with acute myeloid leukemia, a relatively common condition, and performed a genomic analysis on the patients and identified essentially 11 subgroups. And what was interesting is that not only did they have 11 subgroups, but there were many mutations which had an impact on the prognosis even within a particular subgroup. So whilst we may be able to cluster people and say they're in a particular subgroup, they may be suitable for a particular treatment, there are individual mutations will add further granularity and complexity to the prognosis and potentially the treatment of each subgroup. So we're now starting to see the use of new medications in cancer. I'm a respiratory doctor. In my experience, seeing patients with lung cancer in the clinic, through my professional life, it's been very depressing seeing people with advanced non-small cell cancer. This patient's got tumor here affecting the lungs on both sides. This should be black, it's all white. Traditionally, this patient would have had a very gloomy conversation with the doctor about the fact that we had really nothing much to offer, but that we needed to help them make the best use of the remaining months of their lives. Now, by subcategorizing these patients into those that do and do not have a mutation in the EGF receptor gene, we can offer those, as a subgroup of the patients, a new drug called gefitinib. And in those patients who have a mutation in the receptor, it is very effective. And in this example here, all this white stuff in the lungs here has gone away. And this patient has nice black lungs here. So suddenly, we've got a real impact on a daily basis in cancer, where if we can identify patients with this mutation, we can give them a new drug, and it radically alters their prognosis. Perhaps the most dramatic example is shown on this slide. In our world, in Britain, malignant melanoma is a disease that, skin cancer, that is becoming more common. Many people spent a lot of their time sunbathing in their youth and are now presenting in later life with malignant melanoma. Patients with very pale skin are much more vulnerable to this condition. And advanced melanoma, until very recently, was universally fatal within a very short period of time. This patient here, in A, you can see here these little nodules on the skin. This poor person is absolutely riddled with tumor. And the bone scan shows uptake in a large number of metastases all over their body. But this patient had a mutation in the BRAF gene, and this was given a BRAF inhibitor, and very rapidly the tumors disappeared on the skin and the bone scan improved. Unfortunately, after a number of months, the tumor evolved to be resistant to the drug, and the patient deteriorated again. But this really shows us that suddenly we can use this technique, these genomics approaches, not only to subdivide disease, but to open the door to very specific, targeted treatments that actually affect the area that actually affect patients through taking advantage of that particular mutation. And one can imagine over time a series of these, and that combinations, a combined approach, may actually be able to give a sustained remission in a patient such as this. I think another area which is just starting is really interesting in cancer, and that is we can all think about a particular mutation providing a target for a drug. If a cancer mutates, and as a result of that, it's more sensitive to growth factors or something else, and therefore it grows faster, clearly blocking the receptor will be a way forward. But the mutations also, even if they are not functional, are likely to result in the production of abnormal proteins. And of course, it'll only be the cancer cells which have those mutations which produce the abnormal proteins. And those abnormal proteins lend themselves to being developed as targets for vaccines and for the immune system. Historically, that hasn't really worked because the immune system has essentially turned itself off to those new proteins. But new techniques now have been developed which allow us to try and turn on or to reverse that turning off of the immune system. And this is a recent paper where they've been able to activate the patient's T cells and demonstrate that the activated T cells do indeed bind to and proliferate in response to the novel antigens produced by the cancer cells. So this opens another whole new avenue which we can think about developing in the area of cancer care. So not only now are we moving into a position where disease is being subdivided on the basis of the genetics of the person who has it, which is adding huge complexity to the way we manage patients, and it requires a lot more thought and knowledge if we're going to take all this extra new information into consideration when prescribing. But as you will know, because you've done a lot of work in this area, the actual response of the patients is going to be different according to whether whether we give them suitable drugs or according to the genetic makeup of the patient and the pharmacogenomic impact of that drug on the patient. And the study that I like, and many of you will know Sir Munir Mohammed from Liverpool, is where he looked at warfarin. And many of you will know that warfarin is given to a very large number of people, about 500,000 people, I think it is, in the UK are on warfarin, patients with atrial fibrillation, with DVT, with high risk of pulmonary embolism, etc. And that it's a good drug, it stops the blood clotting, and it prevents those diseases progressing. However, it's a dangerous drug. It has a relatively small therapeutic window. If it's given at a dose that's too high, the blood, there's a high risk of bleeding in the intestines or the brain. And so there's a risky period where you have to monitor the INR and get the patient stabilized on exactly the correct dose of warfarin. And what they did in this particular study was to take a group of patients, and in half of them they did a genetic analysis and used that genetic analysis to help them predict the correct dose. In the other half of the group, they just gave them the warfarin following the normal protocols. And what they found was, when they monitored the INR at the start of treatment, the control group, who just had normal practice, there was quite a kick up here. And many of these patients, this is the mean, many of these patients had an INR that put them at risk of bleeding at the initial phase of starting treatment. Whereas, if they had undertaken the genotyping beforehand, they were far better at predicting the correct dose, and far fewer patients got into the danger zone at the start of treatment. So, again, that offers not only a great benefit to the patient, it actually has substantial health economic benefit, because if we look at, in the United Kingdom, 1 in 15 acute admissions are due to problems with medication. So 1 in 15 hospital admissions occur as a result of problems with medication. And on the list of the top 10 drugs that cause those unnecessary admissions, warfarin is high on the list. So if we could prevent those unnecessary admissions, A, we would prevent morbidity and mortality, but we would also prevent the unnecessary costs of hospital admissions to the health service. I just wanted to stray slightly into another area, which is infection. Because this, I think, is an interesting study, and I'm a chest specialist, so I'm interested in tuberculosis. We know that about a third of the world's population have been exposed to TB and are infected with TB. The question is, which ones are going to are at risk of getting active disease? Out of all those who've been infected, who should we actually treat? And this study in South Africa looked at, took blood samples and looked at 16 gene signature within the blood to work out, and were able to show that when they could identify this, a whole series of genes were activated, which indicated that this patient was on a journey to develop active tuberculosis. And when these genes were not activated, the patient was not on that journey, and therefore there wasn't a risk of them having active disease. And so this is a way of starting to think about how we can really target in on those people who need chemoprophylaxis, and those people where we can just say that they're not at risk. What's also interesting, I think, about this area is that there are a whole series of enabling technologies coming along. I just listed 3 up here when I was making the slide, but wherever you go, whenever you go to all the meetings, there are lots and lots of companies coming up with really interesting technologies which will help us to bring this whole area of medicine into day-to-day practice. There are companies now which are starting to develop what is called liquid biopsy, where from a blood sample, where in a patient with cancer, one can take a blood sample, do the analysis, and get the sequence of the cancer cell from leakage of DNA from the cancer cell in the blood, and work out exactly what the mutations are in the cancer cell, and therefore what the next type of treatment you might want to use should be. This is it's not yet in routine clinical practice, but certainly at the recent American Society of Oncologists meeting, there were a number of presentations about it. A number of people now are coming up with point-of-care testing for SNPs to identify specific mutations. This particular one, Quantum DX, was developed by a company in Newcastle and should go on the market later this year. essentially it's a little PCR in a little tiny gadget that you could hold in your hand. It's a cassette, you put it in, and you get a result in about 20 minutes, and it can tell whether you have a particular set of mutations. So the idea of this is to help people actually in the clinic, when they want to prescribe drug A or drug B, which one is going to give you the problems? Let's do the test. Another one that I think a lot of companies are looking at, and that is decision support. This area is very complicated. How can we actually advise doctors sensibly so that they can actually perform their job with modern knowledge, supported by the information that comes out of the genome, and can decision support help them? And certainly in the UK there's a lot of effort on this, because if we're going to get every doctor in the country to work effectively with genomics, A, they need education, and B, they need some support. So one of the missions of our government has been that this whole area of science should deliver benefits to the patients within 3 years. Now that's a very stiff target. So that Genomics England have put together a program to see if they can achieve that. And if we look at the traditional drug discovery model, scientists go into a laboratory, they develop a hypothesis, they apply for a grant, they do some basic science, they do some animal studies, and they get enough evidence to say, well, maybe we should think about going into a clinical trial. Can we take this molecule into man? You've then got, they publish their results, pharmaceutical companies get interested, and they go into phase 1, phase 2, phase 3 studies, and eventually they get a molecule that doesn't cause side effects, and it goes into the clinic, on average 17 years later. And the question is, how do you speed that up? And so Genomics England have created a clinical interpretation pathway with 2,500 medical scientists around the world. The knowledge comes out of whole genome sequencing. We get a sequence for a well-defined group of patients with a particular clinical problem. So we quite quickly get to understand exactly the problem mutation that we need to deal with. They share that widely so that energy across the scientific community can be directed at that particular mutation and develop a therapeutic agent to deal with it. And then because they know the patients in whom it's going to work, they can go back to the specific subgroup and say people with this mutation are the subgroup where this particular drug might work. Therefore, they don't need to do such large clinical studies. They can do them on smaller numbers because it either does or doesn't work in people with that particular mutation. And so the idea, therefore, is that we can move quickly and get agents back to patients within 3 years. To achieve this, the main research organizations are going to play a key part: the Medical Research Council, the National Institute for Health Research, part of the NHS, the Wellcome Trust, Cancer Research UK, etc. So by bringing everybody together, they hope to be able to do this over a 3-year period. So what I hope I've done is given you some evidence that we are moving into a different era. Bruce Keogh, the medical director of NHS England, said, as the microscope and X-rays revolutionized medicine in the 19th and 20th century, so the knowledge of the human genome will dramatically change medicine in the 21st century. We've learned a few lessons. You've really got to spend effort on characterizing the patient's phenotype. We need different methods for sample collection and storage. It turns out that if you want to take cancer biopsies and get whole genome sequencing data, formalin-fixed, paraffin-embedded sections are not a great source of material, and we have to have new sources of material. We really have to focus on the IT dimension of all of this. It's much more complicated than everybody thought. Many diseases are breaking down into multiple different subtypes. We therefore need to think about specific strategies for each subtype. Because each disease is breaking up into subtypes, you need large populations of individuals in order to get enough people with each subtype to be able to do the studies. And part of the reason that we think we can do that in the UK is we have 60 million people in one health system, which is the largest in the world. We're really going to have to emphasize training, not just of staff, but the general public. And we have to keep support of the general public through good governance and regulation. We have a whole range of education programs for healthcare staff, for the general public, and for specialists. And we have an MSc for specialists, which is offered by the majority of British universities and is part online and part through contact with others. So the idea is to make it easy for everyone to participate and to produce something that we can share on a wider scale than just the United Kingdom. When people talk, we talk to other countries about how they might think about this. And as we do have those conversations, there are really a lot of service lines that we need to think about. Many people think, oh, if I built a biobank and I build a laboratory, I've done it. We would say, no, you haven't done it. There's actually quite a lot you've got to do. You've got to change pathways for cancer. You've got to offer pathways of care that are lifelong for patients with inherited disease. You need to think about fertility services, R&D, education and training, regulation, etc. So we think this list is quite important and think that actually there's a role for central and government organizations in planning how they do this in order to get a comprehensive approach. So we in the UK have decided to make a lot of investments in this area. We're on a trajectory. It's going to be a decade or so. We're doing a number of lots of projects, like the 100,000 Genome Project. Results and benefits are going to come out of those. And we're talking to a number of countries thinking, well, what are you doing? We've got a really centralized national approach. What are you doing? Do you have enough people in your population? Do you have enough science and infrastructure? Would it be helpful to think about some form of partnership where you can share some of the benefits of what we're doing and we can work together? And partnership can be of all sorts of types. It could be education and training, or something on a larger scale where a particular country links in with our genomic medicine system, where we have genomic medicine centres, and forms some partnership with those and has all the benefits that we offer the different regions of the United Kingdom. And we have a brochure here which talks about that, which you can see online. And finally, I just wanted to say why I think this is important. If we look 10, 20 years ahead, it's going to be we're going to learn a lot more if we all use the same processes. If we all use the same methodologies, the same analytics, make the same statistical assumptions in the analysis, we're going to get knowledge that is valuable and can be shared between communities. And I think that's the way we're going to get benefit for humankind fastest. And what I think would be worrying is if we all do it in a very different way and we end up with information that really isn't possible to share. So I think there is something about forming benefit. We've created partnerships across the United Kingdom, and the question is, are there opportunities for doing that on a more global level? Thank you very much indeed. I'm very happy to take any questions.
Yes, of course. So anyone has any questions? We'll have the microphones for you. Or there are two. Any questions, please? No? All right, if you have any questions, you can ask from Professor Rory Shore in personal. All right, so thank you very much once again. A big round of applause for Professor Rory Shore. And this is a great information of the genomics and personalized medicine too.